But I'm Annie Lee, and we're from Lacarta Bio, and we're in Kansas City, Missouri. And I'll tell you a little bit about what we do. My talk is going to be kind of vastly different from Dr. Miller's. We come from a totally different perspective. We're helping nutraceutical companies generate early data and publish papers to support their work. So first, I'll talk a little bit about what Lacarta Bio is, a little bit of our background, what we do with some 3D tissue culture, provide some data, and then tell you about three different kind of case scenarios or case studies that we have working with nutraceutical companies, helping them gather data to support and leverage their products. So Lacarta Bio is relatively new. We started in 2012. We spun out of the University of Kansas Medical Center, a diabetes research lab. We have two founders, one of whom is here, Karthik is in the back, and he developed and patented a technology for 3D tissue culture. And that has a lot of possibilities, as a lot of early phase studies are being done with 3D tissue culture as opposed to 2D. So I'll talk quite a bit about that as we go on. So as that company started, we had several applications. This ability to make 3D clusters of many different cells gave us the possibility of becoming a CRO, so a contract research organization to gather data for our clients. We also have our own product where we use the 3D micromold to make cells for transplant as a cure for diabetes in animals. So we actually run our own product as well as products for testing for other companies. So let's see where my pointer is. I don't know where it went. Do you have the pointer?
That's a little skinny one.
Is it up there, skinny white one? Oh yeah, no, I got it. Here? Yeah. So this is a picture of the micromold where we generate the 3D clusters. This is just talking about our contract research and consultative services. And we say consultation because we found, as we partner with nutraceutical and pharmaceutical companies, that some of the early, they have a product that they're excited about. They know that it could be efficacious for patients. They want to get it out to the world. And strategizing about how to gather the data and get that information to doctors and or eventually consumers has been an exciting part of our work. The micromold also lends itself to some other product development, which is another part of Lacarta Bio. And as I mentioned, we have our own product, which is another division of our company. So at Lacarta Bio, we see ourselves as partnering with companies to try to help test products and get them to market using our unique resources. We have a full analytical suite of analytical tools. We all come from different backgrounds. I'm a chemist, Karthik is an engineer, our other founders of pharmacology, physiologist. We have molecular biologists on staff, so we kind of attack problems from a lot of different angles, which has made it really exciting to work with some of the nutraceutical companies that we are partnering with. So to get a little bit into our 3D tissue culture, why is she talking about this? Why are you mentioning this? Well, we find, and it came up a little bit in Dr. Miller's talk, that why aren't doctors embracing this? Why aren't medical schools teaching this in their classes? If there's a product out there that's as good or better or excellent for preventative medicine, why aren't people jumping on this? You would think you could save money, you could help people. Why aren't they? And so our approach is, science talks, right? Do the correct science, do the best science you can, and let science support what you're talking about. Get it into academic journals that are reputable, and do that. So in the pharmaceutical world, the testing has gone from 2D to 3D completely. It used to be if you said your compound had anti-cancer possibilities, you grew those cells in 2 dimensions and you saw if it killed or not. Now that's done. It's all in 3-dimensional cell clusters. And so, why? Because we're not 2 dimensions, right? A 2-dimensional layer is not going to model, say, a cancer tumor or something in our body. I put this in and no one's going to maybe get this, but this is Flat Stanley. Do you even know who Flat Stanley is? He goes around the world and he's paper and you take pictures and say where he's been. I don't know, sorry, it's an internet thing, but we're not flat Stanley. We're not fail, right? Just total fail there. We are not flat Stanley. We're three-dimensional. And so having three-dimensional testing is really important. So I'll go into this a little bit more, but I think that the idea is that we have the ability to really efficiently make three-dimensional clusters so that when you are testing your compounds or actives or whatever, plant extract or whatever you might be talking about, you're getting the best data and the data that's really expected in at least the pharmaceutical world. So this idea of testing not on 2D but in 3D started really in the kind of early 2000s, but probably in the last 5 to 10 years has completely taken off. Just 3D drug screening yielded 1,600 papers on just PubMed. 100 have come out just in 2015. So this is really becoming sort of the standard way of testing compounds. You know, is it killing tumors? What effect is it having on your whatever you're happening to be interested in? So we engineer our 3D cell clusters in a particular way. Again, we patented the technology. It allows us to completely control the tissue that we put in. We can mix different cells depending on whatever you're hoping to test. In a 2D monolayer, it's pretty hard to get different cells talking to each other, signaling in the way that they do in the body. When you pull them into 3 dimensions, the signaling becomes more like what you have. It's more predictive. It's going to give you better data. Our clusters are small, 150 microns, give or take. And this allows your drug or your component or your ingredient or whatever you have, I don't know the proper term for that, but it can diffuse in, it can interact, it can affect the cells that are there. So we have the ability to make a lot of these. They're uniform in size. They're an excellent test for whatever product that you might have. This is just an example to kind of drive the point home. These are just looking at cell number. So oh my gosh, if you're up here, you're 100% alive. If you're down here, this bottom point is saying about 63% are only living. So as you go down, you're noticing that there's a kill that occurs at this particular concentration. So cisplatin is an old chemotherapeutic. It's been used for a long time. And just showing the black circles are testing cisplatin on, I think, ovarian cancer cells in 2 dimensions. The open circles are testing cisplatin in 3D. And so the idea is that if you had tested this in 2D, you would think you have a great cancer drug for this particular cell line because you see a kill starting at relatively low doses. However, in 3 dimensions, you don't get a kill until you're all the way out here. So you're at a relatively high concentration before you're actually killing cells, except if you were basing your data on 2D, you would be giving someone something that's really toxic and it's not going to kill their tumor. So false positive using 2D assays. You could also have a false negative. Fluorouracil in this particular cancer, in 2D, it looks like you don't have a good drug, right? That's never killing the cells. You give it at any dose and they stay alive. Blah, we don't want that. We want those cancer cells to die, which it does in 3D, but we wouldn't know that if we had used a 2D model. So it's really just trying to sell you on the fact that 3D is better than 2D, and it is the way that things are moving. So if you want to speak to doctors or whoever even your public, whoever it might be using the tools that are relevant. So the nice thing about using 3D clusters that we're able to do for our clients or customers, you can use any cell line that you're interested in. If you're saying that this is going to affect a particular disease, you can make the 3D clusters for your initial data. We've done a lot with cancer and diabetes. We've made some from stem cells for chondrocytes and arthritis, stem cells in general, sometimes a mixture of different things. Really it's limitless, and which is why we love what we do, because we partner with and really collaborate with our customers in order to address whatever they might want to see is happening. So you might say, great, 3D, I love it. I'm sold. 3D is great. The problem is, if you've ever gone out to try to do these tests yourself, they're not very efficient ways of making 3D clusters. currently. So if you've heard of the hanging drop plates, if anybody's familiar with that, essentially you put media and the drop hangs down and you seed your cells in there, and then the curve of the drop makes your three-dimensional cluster form. So that's a neat idea, except when you tap the plate against anything and all the drops fall off. Oh, you know, it's a nightmare. I worked with it and it's difficult. So finding an assay or finding a technique to really efficiently and in a controlled way make your three-dimensional cell culture is important, and something that Lucarda has figured out completely. So this is just some hanging drop data. Even in a 384 well plate, noisy data. I don't think you're probably hugely concerned about this, but you can see a tiny bit of a drug curve. This is just cancer being treated with doxorubicin. And you can see the little, you know, your one little three-dimensional cluster that you get in each well. So even if you're 384, you're still having a lot of, depending on what's happening in that well, oh, it knocked off, it's not there, you're not getting beautiful data. This is just our system where we have multiple wells in multiple ability to making these three-dimensional clusters in each well. So you can make 50, 100 clusters per well. You get a nice averaging effect. And this, again, is another talk, another part of our company, but something that we offer our clients, and the data we get is beautiful. This is a picture of so this would be, for instance, in one 96 well, or even one 384 in one well. We're making multiple clusters, and that's what our clusters look like. So it's possible to free them, put them into a different plate if you want to. We're also working on all-in-one assays as another part of Lucarda Bio. There they are, lots of three-dimensional clusters that you can test on. So what's the point? Beautiful data so that you are providing data to wherever you're trying to publish, whoever you're trying to sell on your product. If you're going to a big box, you know, showing data that's relevant. This is the reduction in noise because we have averaged results for our 3D assay. So when I looked up Nutraceutical and 3D cell culture, remember I saw 1,600 hits before and 100 papers alone this year. And I could have been Nutraceutical and cell culture might not be the way to search it, but I certainly didn't find anywhere near the number of papers on PubMed. So, and I think I did Google Docs as well. So the way we see this is just an opportunity. Let's, if we want to provide data that people are going to believe, and you want to speak to the people you want to speak to and make it more legitimate for your medical students, then our hope is always to be partnering and collaborating with in order to get this to the greater scientific community. If you have a wonderful product that is going to affect human beings, let's get it out into the scientific community as a way toward leveraging whatever it is you're trying to sell. So I really only found about 20 papers that was doing this, which was interesting. So now I'll go through. Since I'm talking a little quickly, but now I'll go through a few stories that we have, some of the partners that we've had who have nutraceutical compounds, and some of the things that we've encountered, some of the data that we've generated, and where we're going with that. So first is diabetes, and in the pharmaceutical world, they're interested again in just our ability to really efficiently make 3D clusters. And so we partnered with Eli Lilly to show that. So again, not nutraceutical, but just to give the idea that we know that somatostatin is an insulin secretor, even at low concentrations for humans, but their particular model was native rat islets. So islet cells are the insulin-producing glucagon cells that are found in your pancreas. So here, their model did not show what we know to be true in humans, whereas ours was more predictive. And we published that paper with them, British Journal of Pharmacology. Again, just more data to support that our 3D model and our ability to control our 3D assays was better than what pharma was using at that time. So glibenclamide and glucose, known insulin secretors, we got a better result using our product. In cancer, we have several nutraceutical compounds. We have one there's a plant with what is thought to be a cancer-active compound in it, and how to go about getting the data that they need to either bring this to market as a nutraceutical or potentially take this as a pharmaceutical. And so we grew little mini tumors, three-dimensional, for a variety of different cancer lines. I say other for leukemia and lymphoma. Those aren't going to cluster. Those are blood. Those are just suspension lines that you'll find in your blood. I hope you won't find in your blood, but you get the picture. And just an example of some of the work we did partnering with them is they had this extract, and it was a multi-component, and the question is, what is the active? What are the things we want to focus on? Finding an active out of a plant extract can be really difficult. And so we used ovarian cancer, mini tumors in this case, and just looked at the different components. So we have the ability to do separations in our lab. We looked at these, and it was clear that compound B did not kill any cancer cells. So again, this is 100% alive. And as you increase the dose of their test compounds, you see the cells beginning to die at different concentrations. So the best one is the biggest kill at the lowest concentration of drug is what you're really looking for. So in this case, compound A was looking good. Compound B could be completely eliminated, and so on. So you might say, well, this is pretty, what's the deal? For someone coming with something that they think might have possibilities and working through this with them, it's something that we've really enjoyed doing and was sort of unexpected in our work. Another client had a formulation. They're varying some different natural chemicals and trying to see what the best formulation is. And again, we use our lung cancer 3D spheres in order to look at that. Different formulations, getting it just right before they come up with the product that they want to try to give to patients or people or whatever you would say. So we look at a lot of different formulations for one client. This is a slide that we should talk about because I think that sometimes our clients seem to have things that are a little bit more drug-like. They want physicians to be using these. They want physicians to be realizing that this natural product, this nutraceutical, could kill cancer, could reduce tumor size, could have a therapeutic effect. And so we thought, well, sometimes if we could reduce the amount of classic chemotherapeutic used and supplement with the nutraceutical, you could see sort of either an additive or a synergistic effect. And so could we reduce the amount of doxorubicin needed to kill cancer tumors by adding in some of this drug formulation that is there? And we saw we saw some interesting results here. Synergy is a little bit hard to define, but there is the possibility that the components in this particular extract actually, when combined with certain chemotherapeutics, enhance the effect. So you have something that's not toxic to your body. You can reduce the amount of toxic doxorubicin and see the same effect on that cancer. It's really exciting. I mean, our client was thrilled. We were thrilled. And this came from kind of our pharmacological background, saying you ought to try this with known things. It isn't just that there's nutraceutical over here and then there's pharmaceutical over here and never the twain shall meet. It's that sometimes you might have something that works actually very well in order to reduce something that a patient might need to take that's toxic. So it's just something to think about. Could your product help in that way? I don't know. So this was just a bar graph sort of saying the same thing. You would think if you had known product, I say this is doxorubicin plus your particular thing, that if you just added them together, you might see this much of a kill for the cancer. When actually we saw much more when we put them together. So this was just kind of an exciting result. And we went through different additives different actives, and we do it, this is just a mass spectrum, showing, what the what the molar mass of those might be in order to identify them and that type of thing. But we went on to publish that data with that partner, which was also exciting. So another thing that if you're, so you found this compound and it does something really exciting, and you've done this preliminary data with us, and you've gotten this work and you've published it. Another thing that is people are curious is, how are they working? It isn't just enough to say, this is great stuff, take it, right? No one is going to buy that. Well, they might, but not all will, right? Not everyone will. And so they want to know the mechanism of action. And so exactly how is it working? Is it working on mitochondria? Is it working on the nuclear membrane? What is it doing? And so we also, for our clients, do protein assays. This is just a caspase assay. The black circles show activation of caspase. I should have put the one on there. It's probably 9. It's either 9 or 6. And then the open circles are, this is 100% alive, and then this is the cells starting to die. So you can see as they start to die, the caspase is activated. Again, what proteins are activated? What is the mechanism? How is this actually working? And these are just some different protein assays that we can do. Are these turned on? Are these active? Are these, what's happening if your product is killing something or not? We're in the cancer world quite a bit, and so we're trying to kill cells. You guys are trying to help them, but anyway. And then we compare. We have collaborators who are trying this in mice. Yes, we saw that when this compound, after we saw our data in 3D, they gave it to mice and they did see a shrinkage in tumor volume. And so we're on the road to gathering the evidence toward, for this particular client. And it's going to come out in this journal. So I don't know if you can read that, but haha. Okay, so another company that we've partnered with a Nutraceutical product is Triology. They have an oral rinse that reduces bacterial load in the mouth. And they, by word of mouth, have been actually very successful in getting dentists to use this. It can be a spray, it can be just a rinse, and it's similar to something like Listerine, just to kill bacteria in the mouth for better oral health. But they have a subset of dentists who will not even consider giving it to patients without data, without publications, without something supporting it. The word of mouth is not enough. So they actually have done quite well with their product on the one hand, but they want to open it to the rest. And so this is just a bar graph showing some of the studies that we sort of tried to help put together for them, again looking at cell death. We grew some oral fibroblasts and tested this for them. We compared it to this chlorhexidine gluconate is a popular oral rinse for reducing bacterial load in the mouth. And we looked at some different bacteria and saw that their Triology product was good at reducing those. We partnered with an oral DNA lab to look at, again, different bacteria and the loads. So we worked with them on some human clinical trials. We did some pre and post. We had people rinse with water, and then they rinsed with Triology. And so you're not looking at, what you're looking at is not that these bars seem higher than these bars, but just the difference between black and gray, that there's a statistically significant difference after they rinse with Triology for different pathogens. This is interesting to them mostly, but for you, it's just saying that there are a lot of different types of data that can be generated in order to support your claim. And then again, we published this with them. And so now they use this. They've said it's made a huge difference in their marketing and what they can show to dentists in order to show what they're trying to show. So another thing to consider, and some of you may be in this sphere and some of you won't, but because of our connection to creating three-dimensional islet cells for transplantation to cure diabetes in animals, and our particular geographic location in Kansas City, which is in the animal health corridor, we always think, could this be used on companion animals? And it's not a bad thing to consider. Could what you are producing, could your nutraceutical be used on companion animals? Why? Because there's a huge market for companion animals. By that I mean people will spend a lot of money on their dogs or cats, and sometimes horses. a lot. And that market is real, and we've experienced that. Triology is aware of this. Tooth issue, I will not pretend to be an expert on this, but tooth issues with dogs are an issue. And being able to spray the Triology on dog teeth has been a new market for them. And veterinarians are very much interested in this. It's, how do you get a dog to rinse with something, but being able to spray something that's not toxic, it doesn't stink, it's not super chemical smell, has been a new place for them to go. So we also have vast experience in the animal health world and have sort of tried to help expand our clients into that area. So we just did a quick study looking at bacterial loads in dogs, and not surprisingly, we're able to see on dogs that would let you swab their mouths a significant decrease in bacterial load after the Triology rinse. So hopefully, I've impressed upon you the idea that three-dimensional testing for the early phase work that you might be doing is sort of, where you should be, that there are lots of possibilities, lots of different studies that can be done. I showed a lot of just looking at things dying because we're looking at trying to kill cancer cells, but there are many other possibilities out there. Locardas we're excited about because we're hoping to get it out to the public so that more people can do this themselves. By selling our plates, we hope that you all can do your 3D more efficiently than what's currently on the market. But in the meantime, we're excited about being able to offer this as a service. In addition to that, we have a wide range of different services that we provide, and we've really enjoyed working with our clients. Coming from an academic background, we approach it that way, and we look at how can we figure out a new use or figure out something that you can do with your particular product. So these are just some of the things that we do, and we have some informational brochures out there, and I'm happy to take questions, but I hope that it's given you something to think about. So thank you.
Do you have any questions? Yeah, it's one of them.
Thank you very much.
I don't know, yeah. Right by my
I think you can hear me now.
I think I can.
Okay, so great work, and I personally think the 3D cultures will have a great potential. One question, in these 3D cultures, do you characterize or standardize the cultures, or do you see any similarities between real in vivo situation and
say it again. Do I do what?
do you do any characterization or a standardization to match with the real in vivo situation, or do you at least see the level of expression of some of the markers?
We do look at the level expression of some of the markers. For cancer cells, it varies. I mean, I would say that.
So do you measure cytokines or something like that for that?
We do, yeah. I'm invoking Karthik. Yeah, it depends on our.
Sometimes we will use cell lines because that is the accepted node. Things like head and neck, those are models that have an in vivo version and an ex vivo version. And so the benefits there is those are already well characterized models. We do use a lot of native tissue, so we actually will get, being with the University of Kansas, a human cancer center, and being close to the University of Missouri, they have a veterinary cancer center, so we'll get primary cancer tumors in.
But we have a collaborator at the cancer center who characterizes protein expression for at least our head and neck line. Yeah. So.
And we've done that in diabetes.
Yeah, and diabetes for sure.
And the last question, I
extensively. Yeah.
Do you do also the co-cultures? Do you culture more than one cell?
Yes, we've done it. We've done it with stem cells mixed with, I don't know how much I can, some of it's proprietary, but yes, we are able to co-culture in different ratios and different, to better mimic what's going on.
Great, thanks.
Yep. Yeah.
I've been hearing about this 3D, but now we're hearing also 4D culture, where you allow, where you allow these cells to kind of self-assemble by themselves so that they can mimic more. And they're saying the next
These
10
are
years
go ahead, I'm sorry.
Yeah, because for the 3D, it's mostly scaffolds that, I don't know your model, but
Yeah,
I know
ours
that
isn't.
most- okay.
Ours, you seed the cells and they self-assemble.
Yeah, they're considering that more of a 4D according to literature. I'm not familiar with the 4D.
I haven't
nor
heard
have
4D.
I worked with the 3D.
I don't know what 4D.
But I've heard that the 3D is cell scaffolds, so they're arguing that a 4D model is what most people are trying to switch to.
Then ours would be 4D, because what we don't like about hanging drop is it's confined by the volume of the drop, and you let the drop get bigger, then it gets bigger, and it forces them together essentially. Ours is they're completely self-assembling. It's proprietary how the mold is made, but they come together on their own.
Okay. And if someone wants to test a product, they need to send it to you, or you can send constructs and they work with them, or
Usually they send it to us, and we do it on site. Yeah.
Another question, Becca?
I don't know. Yeah, it seems like it's not on, but then it is. Yeah.
I don't remember the exact year, but in 1994 we had published some data where we got primary cultures from the cancer center and developed spheroids to study invasion. And those studies correlated very well with the 2D cultures. We've never called it 2D cultures, they're called cell cultures.
Yep.
In my talk, I'll be showing some of those studies.
Yeah.
I was just trying to understand that when you compare studies with animals, there's certainly going to be differences because of metabolism, because of different other things. So you will see those differences in any comments on that?
Well, you certainly will see that. I think the idea in any in vitro is simply to try to get as close as you can. And 2D was the standard forever, and it did predict some of it. But probably late 90s, early 2000s, they started seeing some differences, not always, but some, saying that in 2D it doesn't always reflect, and it's clear why. It's just the same reason why even a 3D or 4D, whatever, doesn't exactly mimic what's happening in your body either. Until you're in vivo, you're not going to perfectly mimic it. The idea is just to get as predictive as you possibly can. So in some of our diabetes work, we went back and said, we know what this does in humans, but we do not see that in any culture. We don't see it in, so for instance, rat islets whole, human islets whole, we don't see it. So we don't have a good model. We don't have a good in vitro model for testing for particular diabetes drugs, and we tried to improve upon that, and we were successful in some ways. I mean, I'm certainly not trying to claim or anything like that what we're doing is the end all be all. It's just trying to get closer. And I would say it's becoming more and more accepted that 3D is at least getting closer than 2D. 2D might be fine for some. 2D and 3D will agree a lot, some of the time. The problem is when they don't, I think the winner is usually 3D because it's at least you're having the cells communicate with each other in a little more realistic way than in a monolayer.
I think we got 2 minutes for any additional
One
questions.
last question.
Don't usurp me on the chair.
Good morning.
Good morning.
Usually when I read about cell lines, they refer specifically to Caucasian cell line. And
They work with what?
Caucasian cell line.
Oh.
But cancer progresses differently in different groups.
Mm-hmm.
So, for example, in black men, prostate cancer is more prominent.
Absolutely, yep.
So in the 3D models, is that taken into consideration?
I think it would depend on the origin of the cells. I mean, it would depend on the cell line that we use. It would depend on a variety of things. It's a great question. And I mean, it's the same thing, a lot of the studies that were done on men only and weren't done for women, and that there are a lot of differences there. I think that what's nice is you can control whatever cells you're putting into the three-dimensional culture. You have complete control. So if you wanted to study a particular population, any of that, you absolutely could. If you're going to ATCC, you can see what cells you're getting, you know exactly what their origin is and that kind of thing. I couldn't speak to exactly what our cell lines were, but I absolutely get what you're saying. But I think that you could tailor that and make that work for your, you know, for whatever your application would be.
Okay, if there are no further questions, if you have any other thoughts, you can certainly interact after the next break or at lunchtime. So we appreciate Annie very much for your contributions and stimulating thoughts. And the next speaker is Michael Dodson from Blackmores, Australia, who is going to talk about